Our protocol for high-throughput live imaging of yeast cells enables tracking of growth and gene expression for thousands of microcolonies simultaneously. Differences between strains, between environments, and even between genetically identical cells grown in a shared environment can be quantified. The protocol yields precise estimates of average growth rate and average fluorescence intensity of gene expression reporters. In addition, because so many individual microcolonies are assayed, precise estimates of entire distributions of growth and expression values are obtained.
This protocol follows two main steps: the preparation of the experimental plate and the preparation of the cells for imaging. The cells must be plated immediately after dilution and mixing, so we recommend setting up your experimental plate first. You'll notice the repeated mixing of cells throughout this protocol, which is extremely important in the steps up until plating.
On the day of the experiment, all media and other solutions used for the assay must be filtered with a 2-micron filter, even if they are already sterile, in order to remove any crystals which may appear in microscope images. This includes experimental media and the concanavalin A solution. Next, prepare the microscope plate. In order to keep the glass surface of the wells clear of debris and scratches, always keep a lint and static-free wipe underneath the plate. Use sterile technique on a sterilized bench. Filter 25 milliliters of 1x concanavalin A solution and pipette 200 microliters into each well of the microscope plate. We recommend stabilizing the pipette to ensure proper height.